through a peer review process, be published in an academic scientific journal. He got first place national winner for the science division in the Broadman Masters Science Fair competition, the premier national STEM competition for there's like 10 million students partaking in science fairs across the country, and he's a winner. Prior to this, he was chosen one of the top 30 finalists across the nation for the competition. And he took place in a virtual week-long experience that normally would have been held in Washington, D.C., but was facilitated with Zoom. So he's well versed. Is there anything else you would like to add to that?
No, that's about it. Thank you so much.
Go ahead with your presentation.
Okay, so thank you so much again for that warm introduction. My name is Atreya Manaswi. So I just finished 9th grade, going to 10th grade here at Orlando Science High School in Orlando, Florida. And I'm affiliated with the University of Florida and also the U.S. Department of Agriculture. So I'm going to be speaking today on small hive beetles, specifically the biology and control. And so when people ask me what my research is about, I like to use the phrase, the 3 B's of the beekeeping industry. So what I term this as is bees, beetles, and beer. And yes, you heard me right, that's beer. I'm not insane. I guess we love beer, beetles love beer, everybody loves beer. So coming to my backstory, how I actually got started with all of this. 3 years ago, I met a friend's grandfather and we were on a fishing trip. So while on the boat, we cast out our lines and we weren't getting any capture. So my friend's grandfather began telling stories, and he's about 80 years old. So he is a beekeeper, and decades ago, he told us that he would get dozens of barrels of honey, but that season he'd barely gotten 3. And this was really shocking to me. I remembered the distinct tone of his voice. And after getting back home, I decided to look into this, and I did the statistics and brainstorming. And after 3 successful years of experimentation, I think that I have something great to offer to the beekeeping industry. So I began my research in 7th grade. I've been doing this for about 3 years now. This picture on the top right is me at the University of Florida's Honeybee Research and Extension Laboratory. Yes, I have the same haircut. I was doing a laboratory trial there on pollen substitutes. So I was testing different artificial diets in a laboratory setting and seeing which one was the most attractive to the bees. So in the laboratory environment, it's very comfy, it's air conditioned, there's no stings involved. But I progressed the field trials the years after, and this really taught me a lot about the dedication and the hard work that goes into beekeeping. And I'll be covering this further in my presentation, but it was really eye-opening for me. So the breakdown for this presentation is principally 2 things. So I'm going to be focusing on the biology, the background of the beetle, the morphological characteristics, some behavioral and social things, and some other characteristics as well. And then the second aspect of this will be treatments. So I'll be focusing mainly on chemical-based and organic-based treatments. So these are some takeaways, the things that I'd like you to be able to take away from this presentation. Firstly, what small hive beetles are, what they do to your bees and what they do inside the hives. Thirdly, what I discovered from my research, and 4, how you can apply this in a practical setting, either in your hobbyist or commercial practices. So let's get into it. Beginning with the biology, the scientific name for the small hive beetle is Athena timida, and it was endemic to sub-Saharan Africa prior to 2000. And in Africa, it's not really a threat to the African honeybees there. They're able to defend against this. And if it gets sufficiently bad, then the bees will just abscond entirely. But it was discovered in the US in the 1990s, specifically in our state. In 1998, we got reports from Florida. And since then, it's spread all across the globe, wreaking significant negative impacts on the beekeeping industry. As you know, here in the States, we manage a large migratory beekeeping operation, about 2/3 of our hives going to California each year for pollination. So it's just spread all across the States. And it's reached everywhere. So these beetles are very opportunistic scavengers. They are smart predators of the honeybee, and they destroy honey and pollen stores and damage cappings and comb. And this results in significant stress on the colony. So as you can see on the bottom right, this is an image of a sufficiently bad beetle infestation. And this was something that would cause the bees to abscond entirely from the hive. So here the larvae are defecating in the honey, and this event is known as a slime out. So this is an image of the adult small hive beetle on the left, and on the right, I have a picture of a female and a male. Now, I won't tell you which one is which, but you can actually sex the beetles. At first, you might think that the image on the right here is a male beetle, but that's actually a female beetle. So this image on the left is a male, and the image on the right is a female. This appendage is a phallus on the male, and this is an ovipositor. You see this long appendage on the female. So how you can sex the beetles is actually by pressing on their abdomen, as you see in the bottom right. And one of three things will happen. Either one, a small appendage will shoot out, or nothing will shoot out. And a good rule of thumb is when you're sexing them, if nothing shoots out, then it's generally a male. The second thing that can happen is a long appendage will shoot out. That's the ovipositor, the egg-laying machine, and that indicates that it's a female. And the third thing that can happen is the beetle will explode. And assuming that you didn't do one of assuming you did one of the two things, then it'll be a male or a female, and it won't explode. So this is on the right, a large hive beetle. So there's not a specific species for the large hive beetle. It just refers to an assortment of beetles to different regions. But this beetle you see here, this is endemic to Africa. This is 20 to 23 millimeters in length, and it's called a large hive beetle because in comparison to the small hive beetles, it's large. So the small hive beetles are merely 5 to 6 millimeters in length. On the left are two other beetles that are similar to the small hive beetle that can show up within your hives. And I say this to say that just because these beetles are in your hives doesn't necessarily mean that it's a small hive beetle, and it's not a call for concern. You can reach out to your local bee inspector or just compare based on these images. So this image on the top in the middle here is an example of a sap beetle. So the distinctive characteristic here is these orange spots on the back of the beetle. And this image on the left is a sap beetle. So these are much smaller, 2 to 3 millimeters in length than the small hive beetles. Now, there are 4 primary life stages in the small hive beetle life cycle, the first of which being the egg stage. So here the eggs will develop in 2 to 4 days. The next stage after they hatch is the larval stage. So this is where the larvae will go throughout the hive and they will consume the honey and the pollen stores and damage the cappings in the comb. And the larvae are primarily the root cause for the problems associated with small hive beetles. And after the larvae have developed, they will exit the hive, they'll crawl out, and they'll pupate inside the soil in the vicinity of the hive. So this pupation stage takes roughly 3 to 6 weeks. This is the longest stage in the developmental cycle. And after this, they will emerge from the soil and then go back into the hive and develop into adults. So this transformation into this reddish brown color takes approximately 24 to 48 hours. Now, to put this into perspective, you can see on the lower end, if the beetles are doing well, this 2-day mark, 7 days, and then 3 weeks. This is approximately 4 days from the beginning of the egg stage to the end of the pupal stage. That's 4 weeks. And the life cycle of this beetle is 24 weeks, meaning 4 times 6, 6 consecutive generations of beetles can be within a hive in a single 6-month lifespan, one lifespan of a beetle, if these infestations are not controlled. And actually, one interesting thing to note is that one female beetle alone can lay anywhere from 1,000 to 2,000 eggs upwards in a single lifetime. So it's very important to control these infestations. They can take over even the strongest of hives if they're not controlled. So it's very important to manage small hive beetle populations all around the year. Now, as I mentioned, the larvae are really the root cause of the problem. They defecate in the honey and ruin the honey stores and the pollen stores as well. And they appear suddenly and in large numbers. Now, something interesting to note is that there's this yeast associated with them, this image on the left, called Codema omeri. And so what this yeast does is it initiates the fermentation of the pollen in the honey. When these larvae defecate, it starts that process. And what this does is it emits this volatile called isoamyl acetate. And this is a honeybee alarm pheromone. So once it's released, beetles from the vicinity of the hive, outside of that hive itself, which has this slime out being occurring, the beetles outside of the hive, it serves as like a calling signal telling them there's a surplus of food here. So they'll actually be attracted to that. And during this slime out event, the brood rearing will cease entirely, the queen will stop laying eggs, and the population will just dwindle. So there are really 2 telltale signs of a bad larval infestation. The first thing is rotting citrus odor. If you pop open the lid on the super, it'll probably smell like citrus, something sweet. And the second thing is honey bleeding out from the front of the colony. So this image on the right you can see here is an example of that. And unfortunately, at these 2 stages in the slime out, it's too late to control this infestation. Something interesting I'd like to cover as well is the interaction between the beetle and the honeybee. So what you see here is known as the jailing phenomena. That's not an official term. That's just something I made up. But basically what's happening here is the honeybees are cornering the beetles into cracks and crevices within the hive, almost like a jail cell. So they will rotate around the bees and just guard these jail cells and jail the beetles within them. On the right, you can see here, that's just a stash of beetles there that are being guarded by all of these bees. And something else interesting to notice is that the beetles will actually do something called antenation. So they will take their antennas and rub against the mandible of the honeybees. And they will take advantage and trick the honeybees into thinking that the honeybee is feeding another honeybee. So what this will cause is the honeybee to regurgitate food, and it will actually be feeding the small hive beetle. So this is something interesting that occurs roughly about 10% of the time. The other 90%, it doesn't work, and the bee is not fooled. Something else interesting here is the difference between the wax moth larvae and also small hive beetle larvae. So just looking at it superficially, they do look a little bit similar, but the biggest thing to notice is the size difference between these two. So this image on the top is the wax moth larvae, and this image on the bottom is a small hive beetle larvae. One of the most distinctive characteristics, obviously, with the wax moths is their webbing and cocoons. Something else to notice is their long hairs and also their legs. They have 8 prolegs here in the back and 6 legs near their head, whereas the small hive beetle only has 6 legs near the head and no legs in the back. It also doesn't have any hairs, and it has pronounced abdominal segmentation. You can see here, these humps are a lot more defined compared to these humps, which are rounder. So those are some defining characteristics between these two. Now, these are some good practices for small hive beetle infestations. And just something that's an overarching theme for small hive beetle management is really that it's not one practice that you can implement that will help you control these infestations. It's really the synergy of good management practices and also chemical treatments or organic-based treatments that can help you control for these infestations. So one thing to be careful about is combining or exchanging comb. During this process, you can introduce eggs from outside colonies into these colonies, just small hive beetle eggs. Another thing is with replacing old frames or hives. So introducing dilapidated comb, anything that has cracks or crevices makes for a very good place for female beetles to lay their eggs. And it's also a very good place for beetles to hide in. And the bees obviously can't reach them. Another thing is that when pulling the honey from the colonies, extraction is very important, doing this quickly because the beetles can really get into those caps and they can defecate in there, and this will completely spoil the honey. Another pretty easy thing that you can do is exposing the hives to more sunlight, so just moving them. The hive beetles are really repelled by the sun. It's repulsive to them, whereas the bees don't really mind. They do get a little bit agitated, the bees, when you're dealing with them, but it's pretty beneficial putting the hives in sunlight. And another thing is nucleus colonies, adding new frames or also adding supers. It's important to add supers or frames as the colony grows bigger, because if you're adding new frames or new supers, it's hard for the bees to patrol these, making it easier for small hive beetles to gain access to good points for them to lay their eggs and also to hide and reproduce. Now, coming to the chemical treatments, there are 2 pretty popular chemical treatments that you've probably seen or probably used in your hives. The first of which being Checkmate Plus. So these are the strips that you apply in your hives. The active ingredient in this is 10% comophos. So once the beetles come in contact with this, they simply die. And the other thing is Guard Star. So this is 40% permethrin, and this is applied as a soil drench. So this targets the pupal stage of the hive beetle. So you can see in this model, the comophos targets the adult beetles in stages 1 and 2, and then the permethrin is in stage 5 when the beetles pupate in the soil. So both of these chemical treatments actually, they target the central nervous system of the insect, and they cause the insect to experience tremors, paralysis, hyperactivity, and then death. Now, coming to the non-chemical agents, the organic and biological treatments, this was the focus of my research. So something popular that beekeepers use is apple cider vinegar. So you may have used this or seen this used in hive traps. And this is the beetle blaster trap. This is actually what I used in my research. I'll come to that, but it's filled in there, and what it serves as is basically an attract and kill bait. So this odorous compound is used inside the hive between the frames, and it essentially lures the beetles, and then they drown in that solution. This is also a baseboard trap. There are many DIY traps. These are normally attractant-based, so these can be used pretty universally within these attractant-based traps. This image on the right, third from the right here, diatomaceous earth, you can apply in the vicinity of the hive, and this serves to dry the cuticle of the insect, so it will kill them, the small hive beetles. And the fourth image here on the very right is a nematode. So this can also be applied in the pupal stage. And what this does is these nematodes enter orifices of the small hive beetle, specifically through the mouth, the anus, or also spiracles, and they will go inside and produce bacteria that will consume the small hive beetle from the inside and also release toxins, which will kill the beetle. Now, this is the schematic that I've created for advantages of the chemical treatments, the disadvantages, and then also the organic treatment that I propose. The two biggest things with the chemical treatments are the efficacy and then also the fast results that you experience using them. But there are many disadvantages with these chemical treatments. First of all, they're very unaffordable. You may have experienced this. Their prices compared to some of these organic agents are just astronomical. They also risk soil, water, and environmental contamination. In fact, I believe in soil, the half-life of Comophos is greater than 1 year, and in water it's greater than 30 days. So it can stay there for a long time and have environmental consequences as well. And it also poses health risks to wildlife, aquatic organisms, humans, and the honeybees themselves. There's literature that documents this pretty well. Long-term exposure to these chemicals can cause adverse effects in the honeybees as well. But the proposed organic treatment is affordable and sustainable. I found it to be extremely effective for small hive beetle capture, and it's also environmentally friendly and poses no health risks. So this is the breakdown of my research on hive beetles. I did this for 2 years. The phase 1 of my research was where I tested 7 organic and inexpensive small hive beetle treatments in a field trial. And the second stage of this was developing a novel treatment based on beer. So this was essentially making a refined version of beer. And beer was actually what I found of those 7 organic agents to be the most effective by far. So coming to the phase 1 of my research, I looked at 7 novel agents. These are all organic. Apple cider vinegar as the positive control for this study, mango and cantaloupe puree with boric acid. So the boric acid was used as a 2% concentration to be the lethal dosage for the beetles, dry yeast, and then peanut and grapeseed oil, the scented oils, and then also beer. So you may be wondering why these compounds were picked. These are all odorous compounds, and the literature has shown that it's documented well that these beetles can fundamentally feed and reproduce on tree sap, rotting fruits, and such odorous things. So that's why all of these compounds were chosen. And specifically fruit, there are papers that show that the beetle can feed and reproduce well upon fruit, such as apples, bananas, and other things. And this image on the right is an image of the beetles being trapped within the small hive beetle trap. These are dead beetles. So I ran an extensive field trial, and each week I would fill the beetle blaster trap with the treatment, and I would place them in an upper super and a lower super within the hive. So this was done for 3 hive sets per treatment, and I used a total of 24 honeybee colonies, which is over 2 million bees. So you can see on the left, that's me in my beekeeping suit. You might be thinking, I'm wearing the gloves, I have the beekeeping suit on, I had a smoker, what a young, responsible, good beekeeper, what a responsible young man. But no, that's wrong. I was a horrible beekeeper. The first time I got into the field, I was stung so many times, dozens. I mean, what type of beekeeper are you if you haven't been stung, right? But I really experienced that in the field. And it was painful. So after each week, I would extract the traps and then I would do my beetle tabulations. So I did this in my mother's kitchen during the pandemic in 2020. And often the beetles would get loose in the household. She wasn't all that fond of this idea. They'd fly around and my brother and I would have fishnets in hand, chasing these beetles around the house. So that was very fun. Now, this was my stats analysis. So after that extensive data collection and 6 weeks of field trials, I did stats analysis. So this is a t-test, and being in middle school in the 7th and 8th grade, I couldn't tell my mouth from my behind, and I didn't really know exactly what I was doing with my statistical tests, but I came to learn a lot from these stats tests. And basically what I was doing here was comparing apple cider, which was the positive control that I was using in the study, against each of the treatments that I had sampled. So here you can see this first column is apple cider versus mango, and apple cider versus cantaloupe versus yeast, peanut, and etc. So These green arrows are showing the significance for each treatment. So the treatments, essentially what I'm doing here is comparing apple cider and the other treatment and determining if the treatment that I'm comparing is better than apple cider. So mango mix was better, cantaloupe was better, yeast was better, and beer was better. These 4 treatments were better than apple cider. And the next step in this stats analysis was running an analysis of variance. So what I was doing here was comparing the means against one another to see if there was a significant difference, basically if there was an outlier from the data set, something significantly better than the other treatments. And this is given in the form of a yes or no response, a significant difference or a non-significant difference. And I did find a significant difference, but I didn't know which one. So I took this further with something called a Tukey's test. And so what this identifies is which actual group is different. And so what I found from this was beer was by far the best group for small hive beetle capture. And at the time, I didn't know about charts, graphs specifically, and these are a very, very good way to visualize this. But this is the Pareto chart, and I have all these red lines here, but there's something cool about this Pareto chart called the 80-20 rule. And basically, if you start at this 80% mark, you draw a line here and draw a vertical line down, and then 20% of the segment on the left, which is in this red circle, accounts for 20% of the entire population. So you can see beer in dark blue here compared to the other treatments, specifically beer versus the apple cider here in this other shade of blue. This is significantly more effective than apple cider, and beer makes up for a majority of the beetle capture, showing the significance of this treatment. This is the box plot, another way to visualize this. So the X you see here, this circle, represents the mean, the average capture of beetles using this beer treatment. And it's significantly higher than any of the other treatments here. So what I found for my phase 1 of my research was that beer was 18 times more effective in beetle capture than apple cider vinegar, and it was the best treatment by far margin. Moreover, beer is very inexpensive and it costs merely 5 cents per hive on average. It's also widely and readily available, and it does not harm the bees, only the beetles. In fact, the slits on the tops of these traps are small enough so that only the beetles can come inside these traps, but the bees do not interact with them. Some literature has also shown that if the bees are aggravated by this, they'll cover the trap with debris, and that's if they're aggravated by it. But I did not observe that in my field trials. And now I mentioned the cost, but this is just something that I wanted to highlight here. These are just screenshots from walmart.com that I found. You can see here, this is the beer that I used, the Miller High Life beer. And this is 7.3 cents per fluid ounce compared to apple cider vinegar, which is 19.8 cents per fluid ounce. So this is almost a third of the cost. Beer is a third of the cost of apple cider vinegar. So it's extremely affordable. It's much more affordable than apple cider vinegar, applying this on a large scale, and it's also much more effective. So it's a third of the cost, and it's also 18 times more effective than apple cider. But I wanted to take that monumental finding that beer was extremely effective a step further in the phase 2 of the research. So this research focused on the development of a novel blend based on beer's chemical composition. So this was essentially a refinement of beer, and the goal of this project was to create an even more effective and even more affordable treatment than beer. And so how I went about doing this was by collecting the chemicals from beer, and these are referred to as volatile compounds. So these are compounds that just evaporate readily at room temperature. So once collecting the volatile compounds inside a trap, I placed them inside this unit. This is called a GCMS. So these adsorbents were analyzed via GCMS, and this analyzes the chemical composition and breaks it down. So each of these peaks on the right, this is the image on the right is called a chromatogram, and this is what is produced from this instrument. So each of these peaks represents a chemical, and this was basically analyzed, and the chemicals that were on the left side of the spectrum were chosen. So these chemicals on the left were chosen because they weigh less, and that means that they're more easily carried in the air. They have a greater volatility, a higher vapor pressure, and this means that they're more easily picked up by the beetle, and they're picked up first. So it was these compounds likely that were making beer attractive to the beetles. So the goal of this project was to concentrate those compounds to make an even more attractive blend for the beetles. Now, in addition to the GCMS analysis, EAGs, electroantennagraphies, were also performed. And this is basically just taking the beetle antennae and seeing what the beetle antennae's response was to those specific chemical compounds. And so this image in the middle you can see here is the hive beetle under the microscope. And this is like neurosurgery, but on hive beetles. These beetles are very, very tiny, 5 millimeters in length. So it's very tricky work trying to excise these antennae from the beetles' heads. And these suckers, it's almost like they didn't want to die. I mean, how dare they? They just didn't want to die. So we actually had to put them inside Eppendorf tubes and ice them. And at that point, they froze, and we were able to take the antennae off from these beetles for the analysis. So this is the setup for the EAG here. And basically, I was testing to see whether these antennae would have a response to the chemical compounds that they were being exposed to. And then based on that, the blends were formulated. So there were 4 treatments that were used in this trial. There was an oil blend, an oil water blend, beer as a positive control, and then also a blank trap. So those are the 4 treatments used in the field trial. And again, I used 28 hives, and they were used at 3 different apiary sites for 8 weeks during fall. And so this is an image of the trap you can see with a very high beetle capture on the right. And again, more numbers. You're probably sick of this at this point, so I'll try to be quick with this. But basically all this was telling me was that the oil blend was the best treatment by far. I ran about 12 confirmatory tests to document this well. So again, figure A here, the ANOVA, showed that there was a significant difference somewhere. I used the Sheffy's post hoc test, figure B, to identify which group was the best. And the oil blend was by far the best group. It was in fact shown to be 5 times more effective than beer. And then figure C is called type 1 and type 2 error testing. So this is another analysis that I used to make sure that I was running all of my stats correctly and making sure that I had no false positives or false negatives within my data set. And so this type 1 and type 2 error testing validated the previous statistical findings. And so the oil blend was by far the most significantly different group. And this is a good way to visualize it without any of those numbers. The oil blend here, which was shown by the stats analysis to be the best, is in gray. And you can see beer here in this mustard color. So the oil blend is 5 times more efficacious than this beer. Again, using that 80-20 rule from earlier, you can see, drawing a horizontal line here and a line down, that the oil blend makes up 20% of this, makes up for the entire population, essentially. So it was very significant compared to the other treatments, the oil blend. So what I concluded from this was that the oil blend treatment I found was extremely effective by a far margin, and it was shown to be 5 times more effective than beer and half as expensive. Okay, so this is why you should use the findings from my phase 1 research. Unfortunately, unless you have access to a GCMS and an EAG, you won't be able to use the volatile blend, but you can use beer. So beer is a great treatment because it doesn't harm your bees, firstly, and it can be used in any in-hive trap as an attractant. It's also extremely cost effective, as I highlighted earlier. It's 1/3 of the cost of apple cider vinegar, which I'm sure some of you may use. It's a popular treatment or an organic-based treatment used by beekeepers. And in fact, if you're not using the beetle blaster traps, any of those DIY traps or the baseboard traps which use some sorts of oils are based on attractants, and you may be using apple cider vinegar or any other scented oils, but you can try using beer. It's extremely cost effective. So if you give that a shot, it may work very well for you and help you control your beetle infestations. So my key takeaways on small hive beetle management would be, the first thing is having good apiary practices, maintaining a clean apiary, giving your hives good sunlight exposure, also being careful with adding the supers and extra comb, extra frames, so that the bees are able to patrol the hive. Use of treatments to control small hive beetle infestations. Specifically, I am a big believer in the organic treatments, but using either an organic-based approach or a chemical-based approach to treat for the hive beetles. And ultimately, it's the synergy of these two that reduces beetle populations to a low, manageable level. Now, these are some resources that I have for small hive beetle management. This is from UF. This is the second link is something about small hive beetle biology and also some helpful things that you may find useful. And the third thing is the UF page. They have some good videos about small hive beetle management practices there. So I'll share this and hopefully this can be circulated and you guys can use this, and it's pretty helpful. So I definitely recommend checking this out. And so that's all I have for today. Thank you so much for putting up with me and my horrible sense of humor for the past half an hour. I really appreciate it. So thank you for your time and attention. And I'm open to questions now.
I'm going to take questions.
You like me?
Ask a question.
Okay.
Can you hear me?
Yes, I can hear you.
So you say that we don't have access to the formula you use. Could we mix oil and beer and get a good result, or would that be a bad thing to do?
Okay, that's a good question. Thank you. So you could use beer, but essentially what I was doing in this study was a little bit different. I was refining beer. And so it wasn't just beer, it was a concentrated version of beer using specific chemicals from the beer. So the best thing that I can suggest is using beer within the hives, and it works very, very well. It was shown to be 18 times better than the apple cider. So it's definitely an effective treatment. But unfortunately, you can't formulate the oil blend unless you have access to a GCMS. So.
Another question.
Have you heard anything of using peppermints inside the hives to eliminate the beetles? I've seen a lot of videos of that online lately.
I haven't actually seen anything about the peppermint, those essential oils for the hive beetles. I have seen things about those essential oils being used for the varroa mites. I think they work pretty well against the varroa mites. Sorry, what?
Using the peppermint candies, not essential oils. And
oh, peppermint candies.
Yeah.
No, I have not.
To the bottom of the house. Okay.
No, I have not heard about that. That's very interesting. Sorry, I don't know about that though.
More questions, back
What do you
here.
put the beer in?
Okay, so the beer, yes. So the beer is placed inside these in-hive traps. You can fill about, I use 10 milliliters inside these 10 to 12 milliliters. It's about halfway. And so all you have to do is place about 12 milliliters of the beer in there. You can just fill it about halfway. It doesn't have to be 12. It's just enough so that the beetles are able to fall inside that trap without just taking a sip and then leaving. They fall in it and then they drown. So that's why halfway is used. So you can just fill it in the trap through the slits and then put it inside the hive.
I have one question for you as well. Have you looked at that malicious earth filters used for beer and selecting the beers to see which one has more non-malicious earth in there and the potential effect on the beetle that way?
I'm sorry, I didn't hear the question quite clearly. Do you mind repeating it?
Those beer manufacturers use a diatomaceous earth filter somewhere in the process of making their beer. The different brands have different chemical compositions. Some of the filters will actually filter out more of the chemicals that you might be looking for than the others, because they may use that for taste quality in the beer. If you've seen a taste tester for beer and you could apply that to what the beetles go for, you could look at the different beer solutions to potentially gain the taste that the beetle is actually seeking. And it might improve your overall gain.
Yes, that's something interesting. I actually used, I was just using one brand of beer. I used that brand specifically, if you may be wondering, just because it was inexpensive and readily available. I found it at Publix, and that's why I was using that brand specifically. I did not look into a beer comparison study, but that's something definitely interesting that could be looked into future research. So thank you. I see a hand raised.
Question I have is, anytime you add an attractant in a hive to lure in hive beetles, do you feel like your hive numbers are because you're drawing in more beetles from the environment for that thing? And is there too much attractant that would cause actually an ill effect because you're actually intentionally luring beetles from the environment into the hive?
Generally, this serves as yeah. So generally, this serves as Okay, so this normally this serves the in-hive traps serve as just an in-hive control measure. Normally the odors of this aren't strong enough for beetles from the environment to be attracted. It's normally just the beetles that are already existing inside the hive that are attracted and lured by the beer compound in this case. So
On the hive beetle numbers were reduced significantly by the trapping method.
yes, in-hive trapping is in-hive trapping is a very effective method that you can use for trapping the beetles. Using these types of traps, you can also use the outside measures, things like the diatomaceous earth, or also the permethrin spray. Those are things for outside of the hive.
Thank you.
I see one hand raised in the Zoom chat by Desiree.
Hey there. Yeah, thank you. So number one, I'd like to just say your research is very impressive and I appreciate it and appreciate you sharing this with us tonight. But I had a question about the diatomaceous earth. I've been kind of hesitant to use it in the apiary. I've used it other places, for ant control and stuff, but I was afraid that if I spread it around in the hives, it could also hurt the bees that, as they land on the ground. Is that not a concern?
So generally the bees actually, they can use their wings and they'll actually vibrate, and the diatomaceous earth will oftentimes just fall off. The dust will fall off of them. But something to be careful with is especially just using it on the ground and avoiding putting it near flowering plants. That's the biggest thing, just using it in the vicinity of the hive and being careful with that. I don't think it's extremely toxic to bees. Something like permethrin is something that's very toxic to the bees, but something like this, you shouldn't have too much concern if you want to use it. You just have to be careful not to, put it onto plants near the hive. That's the biggest thing.
That makes a lot of sense. Thank you. Appreciate that.
Of course. Are there any other questions?
This one's way bad.
Will this be posted on the website? Is your presentation to be posted on the UF website?
Oh no, this isn't going to be posted on the UF website. I do have my email here. I can share the details if you'd like. If you have any questions, if you'd like to reach out, I can definitely share some helpful links with you. But no, this presentation itself isn't going to be shared there. I do have these resources that I spoke about earlier. I'm not sure who the right person to share this with would be, but I can drop these in the chat if you'd like. These are some helpful things that I touched on upon my presentation, but this can definitely be helpful for beetle management.
Send them to you.
If you'll go ahead and send them to me,
Sure.
then I can share with the club.
Okay, I'll do that.
Science magnet scope.
Yeah. Okay. Anything else? All right, we'll say goodnight then. Thank you.
Thank you.
Thank you very much. Awesome presentation.
Thank you so much. It was such an honor speaking here. I'm so happy. Hopefully I can come back again. It was really fun. So thank you.
Very impressive. Good luck.
Okay. Hope you have a good night then. Bye-bye.
Okay. I thought he was quite impressive. I saw him a little bit of the preview.